Bonsoir tout le monde. Si vous voulez bien prendre place, on va démarrer notre soirée. Bon, Madame la députée, Monsieur le député, Monsieur le Président, Mesdames et Messieurs les membres de l'Assemblée, les membres du Conseil d'administration et les membres du Conseil scientifique de l'Institut Pasteur, Mesdames et Messieurs les membres du jury, chers collègues, chers amis. Louis Pasteur serait probablement fier de nous voir réunis aujourd'hui pour célébrer la recherche scientifique, 200 ans et 30 jours après sa naissance le 27 décembre 1822. Je suis heureux d'ouvrir officiellement la cérémonie de remise des prix scientifiques du bicentenaire. Ladies and gentlemen, my name is Stewart Cole and I am the Director General of the Institut Pasteur. It is my pleasure and indeed a great honor to welcome you all to the Institut Pasteur for this special event that officially marks the end of the celebrations of the bicentenary of Louis Pasteur's birth in France and around the world. When I decided to create these prizes 1 year ago, I said to myself, what better tribute to a global figure in scientific and medical research than a scientific award that bears his name? The Louis Pasteur Bicentenary Prizes reward scientists who have distinguished themselves by achievements reflecting l'esprit Pasteur, the Pasteur ethos, in the fields of biomedical research, public health, or innovation. 5 scientists, female or male, consolidators or advanced in ERC parlance, and of various origins will soon be revealed. They come from the Institut Pasteur, the Pasteur Network, and from our alumni. They perfectly showcase Louis Pasteur's claim that science knows no country because knowledge belongs to humanity. Indeed, I am fully confident that Louis Pasteur would be very proud of their scientific work and their contributions for the benefit of humanity. The total value of these prizes is 200,000 euros, which is, and here you will surely agree, more than symbolic. This generous amount was fully funded by the Carlsberg Foundation in support of excellent scientific research and in tribute to Louis Pasteur's own work on fermentation. I traveled to Copenhagen in Denmark in October last year for a splendid symposium on his legacy, organized by the Danish Pasteur Society. This event confirmed my conviction that the relationship between Louis Pasteur and Jacob Jacobsen, the owner of the Carlsberg brewery, was both close and meaningful. Today, it is my privilege to welcome Professor Birgitte Skardhauge, Vice President of the Carlsberg Research Laboratory, and Mr. Anders Reed, Managing Director of Cronenbourg, France. On behalf of the Institut Pasteur, I wish to warmly thank you for your generous support. So please join me in applauding our sponsors and friends from Carlsberg. I would also like to take this opportunity to salute and thank all the Institut Pasteur's partners and friends, of which the following is a non-exhaustive mapping. Now, I'd like to thank the members of the international jury, chaired by Professor Pascale Cossart, Honorary Permanent Secretary of the French Academy of Sciences, who've all made the trip to Paris for this ceremony. I will let Pascale introduce this very prestigious panel of 13 scientists, including 2 Nobel Prize laureates. Many thanks to all of you. My final words are for the winners, whom we are about to discover, to congratulate them for inspiring us. My objective in launching these bicentenary prizes was to promote excellence in science and to encourage and support future generations of scientists. In that sense, this event is very much oriented towards youth and towards the future of science. 200 years after Pasteur's birth, the world needs scientific research more than ever to meet the great challenges of today's world, as was so stunningly illustrated during the COVID pandemic. Let that lesson be remembered. Louis Pasteur would surely be immensely proud of the new generation of scientists whom we are celebrating at this event. Many thanks to everyone involved, and now Birgitte and Anders, the floor is yours.
So thank you very much, and I must say it's a great honor for Anders and I that we can be here today, and especially also here to be in the famous Pasteur Institute to celebrate the 200 years anniversary of the birth of Louis Pasteur. In Carlsberg, in the Carlsberg laboratory where I work, I have been working there for 30 years. Every morning I have actually said good morning to Mr. Professor Louis Pasteur, because we have a very nice monument of him at the entrance hall. So that's also to show the big respect Brewer Jacobsen had for Louis Pasteur. Also above the famous Elephant Gate at the original Carlsberg breweries in Copenhagen, sorry, the golden world. Oh. Yeah, I think I need some. The golden words of Brewer Jacobsen state that Carlsberg should always strive for the highest possible quality in everything we do, and never focus solely on immediate gains, but also give thought to the future. To achieve this ambition of perfection, Jacobsen relied on groundbreaking science and innovation. And in 1875, he established the Carlsberg Research Laboratory, which soon produced significant scientific results, not only to the brewing world, but also to discoveries and to society in general. Through these works, J.C. Jacobsen helped to ensure that Carlsberg today, but also many years later, will be one of the largest, or the world's largest brewing groups, owning some of the industry's strongest brands. In this audience here today, I do not think there is any need for explanation about Professor Louis Pasteur's contribution to the industrial microbiology and medical science during this time. And I probably do not need to say anything or to clarify to you about Brewer Jacobsen's strong admiration for Pasteur, which was very considerable. In the 19th century, brewing saw great technological innovations: steam, cooling, bottling, and a lot of machinery. But quality control remained still very problematic. So Etude au la bière from 1876 was Jacobsen's Bible. In this study, Pasteur demonstrated that yeast used for beer production contained impurities, and he tried to clean it with tartic acid and also with alcohol. The method worked somehow, but it was not really 100% perfect, and many brewers still had quite a lot of problems with production of good quality beer. So the Danish microbiologist and fermentation physiologist, Professor Emil Christian Hansen, whom Jacobsen had employed for his new Carlsberg laboratory, was also a very passionate student of Pasteur's work. Hansen took Pasteur's groundbreaking discoveries to the next level by demonstrating that yeast is not a uniform concept or substance, but it can be divided into a number of different yeast types. Those suitable for brewing, he called cultivated yeast, and the remainders he called wild yeast. Hansen developed a method for the separation of the different species of yeast, and in 1883 he purified the first yeast called Saccharomyces carlsbergensis. And he also designed the first yeast propagating apparatus for cultivating pure yeast strains. So based on Pasteur's discoveries, Hansen's scientific breakthrough and technological development revolutionized the brewing industry and the whole process, and it was made available for the international fermentation and brewing industry. Brewer Jacobsen's respect for Pasteur was limitless, less so for his own Professor Hansen, although people referred to him as Pasteur of the North. Pasteur himself played a significant role in easing the relationship between Jacobsen and Hansen. So in August 1884, Pasteur arrived in Copenhagen with his wife for an international medical conference. Jacobsen quickly invited them to be his personal guests, and Pasteur paid several visits to the brewery and also to the laboratory where he met Hansen. The two scientists discussed and exchanged their theories, and Pasteur invited Hansen to come to Paris the following year, where he was introduced to Pasteur's laboratory and also to meet his colleagues. Hansen's respect for Pasteur was indeed returned, and in 1886, Pasteur awarded Hansen the gold medal of the Society of the Development of National Industries here in France to Hansen. So when Hansen in 1901, at the end of his career, summed up his own work, he made Pasteur's famous saying into his own words. There is nothing more fulfilling to a scientist than to make new discoveries. But the pleasure is doubled, however, when he sees direct use being made of them in everyday life. So the Carlsberg Foundation is one of the world's oldest industrial foundations. It was established in 1875 by Brewer Jacobsen, and it is today the controlling owner of Carlsberg AS, and it gives back to society by significant support of science, arts, and culture. As mentioned earlier, the Carlsberg Foundation was founded on passion, a passion for perfection. The Golden Words also provide the platform for the Carlsberg Foundation to provide funding every year for the finest research, science, and art, and to play an active role in the efforts to overcome some of the world's global challenges. So, in connection with the 200 years anniversary and celebration of the birth of Louis Pasteur, and to celebrate Professor Pasteur and the power of science, it's a great pleasure for the Carlsberg Foundation to support the Louis Pasteur bicentenary prizes for achievements that capture the Pasteur ethos. So, merci. And Anders, I think you will continue.
Yeah, and as you could see, I had to give Birgitte a glass of water. I'm sure she would have preferred a beer, Birgitte, Carlsberg. So that will have to be next time. So I'm then the commercial colleague of Birgitte. So thanks to you all. So Carlsberg, actually, then in France, and for many of you French, you know probably better than Brasserie Cronenbourg, a company that actually has a history back to 1664, and also then 1664 being the year where Brasserie Cronenbourg, so Carlsberg then in France, when it was actually founded, and also then the name today then of one of the fastest growing beer brands that you have globally, C64 Blanc, which is the fastest growing beer brand then in countries like China, Vietnam, and also where I come from then in Northern Europe from Norway. And Brasserie Cronenbourg is actually also then possessing then the biggest brewery in France, not far away from Strasbourg, in a small place called Obernai, a beautiful little village. Carlsberg is very engaged in the French market because it's actually one of the most interesting ones in the world. The French consumer is drinking more beer and less wine, actually, year on year. The French are also enjoying more and more expensive beers, hence a very interesting market also then for us commercially. For us, that means also then offering a lot of different beer brands like Carlsberg, like Grimbergen, like Brooklyn, La Bête, and some of these brands that you might know. But it's actually also very important for us in the spirit of J.C. Jacobsen to continue to evolve and to innovate. The maybe most substantial effort for us as a brewer in France, and also then to contribute to a lower consumption of alcohol among the French, is to transform the market, so the beer market, and from alcoholic to more non-alcoholic beers. And Carlsberg in this context is a clear market leader globally, but also then locally here in France. And I know that maybe some of you French in the room here, you've tasted this brand called Tourtel, which you might not think is a beer, more like a soft drink, but it is actually a beer, and it is purely brewed as any other beer. I'm just my final kind of closing remarks from myself and from Birgitte is that, of course, we will then have to offer you some beers tonight. They are with alcohol, though, so it's not a tourtel. And you will find this classic 1664 blonde. I think they're back in the hallway somewhere after the session. And I guess you've tasted that one before, but also we have one that I don't think you have tasted before. And I'm sorry for being such a highly commercial person, but it's after all, it's my job. And this is a 1664 Blanc Prestige. It's a quite unique thing, actually. I would be very surprised if you ever tasted it before. So this is a 16 Blanc, but it's made of champagne yeast. And it's very much then in the spirit of both J.C. Jacobsen and Hansen and Pasteur, I would say. So please have a sip and taste it afterwards. You might have some wine as well if you insist, I'm sure. Take the opportunity to enjoy it. Actually, this product, you will only find it in China because it's brewed in France with this very special yeast, and it's only sold and offered to Chinese customers, and then probably some very high-end and super premium ones, for what I know. So by this, Birgitte and I would like to very much then thank you for having the opportunity to speak to you all then about maybe in this context a subject a little bit different. But anyway, thanks a lot for listening, and both Birgitte and I wish you an excellent evening, and especially with the happening that's going to take place now in a few seconds. So enjoy the evening, and let's meet for a beer maybe after the session. Merci beaucoup.
Soyons sérieux maintenant. Okay. I was told to speak in English, so dear Stewart, dear Professor Birgit Scott-Auge, dear Professor Underrode, dear distinguished guests that I unfortunately cannot individually name. You are too numerous, but I sincerely thank you for coming to this ceremony. Dear Pasteurian colleagues, dear friends, I am extremely pleased and honored to see so many people attending this ceremony of the Pasteur Bicentenary Prizes. It really means a lot. It means a lot for Stewart and his team, for me, and for the whole Institut Pasteur. So for those of you who do not know me, I am Pascale Cossart. I am the Secrétaire perpétuel honoraire of the French Academy. I am professor at the Institut Pasteur, where I have been most of my career, more than 50 years. And I am now a visiting scientist at EMBL Heidelberg. I was the president of the international jury. So when Stewart Cole first contacted me to discuss about his wish to honor Pasteurians with prizes which would be given to close the year celebrating the Pasteur bicentenary of his birth, I have been immediately enthusiastic about the project. When he asked me to be the president of the jury, I accepted immediately upon some conditions. I would be the one to choose the jury members, and the jury would be totally independent from the direction. Stewart accepted without any problem. In addition, I asked to be helped by someone who had been working with me before, and I am naming Caroline Bayergeau. This was really accepted. Caroline, I have been really, really pleased to work with you. She has been amazingly efficient and nice. Caroline, I want to publicly thank you for all your dedicated efforts among the last few months. I must admit that all the high-profile scientists that I have contacted responded extremely rapidly and positively, except two whose competence might have been anyhow a little bit far from the topic of the prizes. So as you see on the slide that will be presented, the international jury was composed of 13 members of various backgrounds, competence, and coming from highly prestigious institutions. It has been a great privilege for me to work with such a distinguished panel and a really committed group of scientists. Today, 7 members of the jury, including me, are present for this ceremony. So Professor Françoise Barré-Sinoussi, Nobel laureate in medicine and physiology in 2008, professor at Institut Pasteur, honorary president of the Pasteur Network Paris. Merci Françoise. Professor Emmanuel Charpentier, 2020 Nobel laureate in chemistry, director of the Max Planck Unit for the Science of Pathogens in Berlin. Thank you, Emmanuel. Professor Philippe Sansonetti, emeritus professor at the Pasteur Institute at the Collège de France. Philippe, merci. Professor Alain Fischer, honorary professor at the Collège de France and founder of the Institut Imagine in Paris. Merci, Alain. Professor Jeff Almond, global head of research at the Sanofi Pasteur from 1999 to 2013, visiting professor of microbiology at the University of Oxford, UK. Thank you very much, Jeff. Professor Staffan Normark, Karolinska Institute, Sweden. Thank you, Staffan. So please join me to warmly thanking them for their time of contribution. We will upload the laureates later, but I would like to applaud the jury member. So as Stewart said, the Louis Pasteur bicentenary prizes aim to reward scientists who have been distinguishing themselves by achievements reflecting the Pasteur ethos in the fields of biomedical research, public health, and innovation. So 5 scientists from the Pasteur Institute, the Pasteur Network, or the Pasteur alumni community will be presented by a member of the jury. So the senior scientist from the Pasteur Institute will be presented by Philippe Sansonetti, the junior by Jeff Almond, the Pasteur Network Prize by Françoise Barré-Sinoussi, the Junior Pasteur Alumni Prize by Professor Staffan Normark, and the Senior Pasteur Alumni Prize by Alain Fischer. We received a large number of applications. Applicants had been asked to submit a 750-word summary describing one achievement supported by a link to a seminal publication, a clinical trial, a device, or a patent. They had to send also a short CV and their 5 most significant publications. The selection process was organized in 2 steps. Each application was first reviewed remotely by 5 or 6 different members of the jury who gave A, B, or C. A to be retained, B to be discussed, C not to be taken into account any longer. I must admit that this first step worked amazingly efficiently. We ended up with 3 or even 2 candidates for each category. A big table with all the evaluation was presented to all members anyhow. Then all members of the jury met together by video conference and selected the winners for each category. Discussions were quite animated, but decisions were obtained after taking into account all opinions, including the opinions of 2 members who could not be present but had sent their comments. Congratulations to the laureates for their work and achievement. I agree with Stewart, Louis Pasteur would have been very pleased to meet such personality. So before starting the ceremony, I wish to thank again our sponsors, as well as the staff who has been involved. I already mentioned Caroline, but there are many others, including Louis Marty, Valérie Escandre, and the many other members who contributed to this event. So thank you, all of you. I will now let Christophe Denfer. Professor Christophe Denfer is the Senior Executive Scientific Vice President of the Pasteur, and he's going to launch the ceremony. I wish you a very nice evening.
Thank you, Pascale, for introducing the prize, the process, and thank you very much for all the work that you and the jury have made to reach decision on these 5 prizes. Just to remind you that there are 2 prizes that have been awarded to a senior scientist and a junior scientist working at Institut Pasteur. One prize awarded to a scientist working in the Pasteur Network. And finally, 2 prizes awarded to alumni, a junior alumni and a senior alumni of Institut Pasteur. So the first prize that will be awarded tonight is going to a senior scientist working at Institut Pasteur, and the prize will be presented by Professor Philippe Sansonetti. So Philippe, if you want to join us. As Pascale already said, Philippe is an emeritus professor of the Institut Pasteur and Collège de France, and is also a member of the French Academy of Science, the Royal Society, and the National Academy of Science, and well known for his work in cellular microbiology and pathogens vaccine development.
Thank you very much, Christophe. I'm very honored and delighted, actually, to be here and to introduce our senior scientist Louis Pasteur Prize of this year. I don't know which be the next year of these prizes, but I won't probably not be there. So I think the best thing we can do is first start with the video, which summarizes the career, the brilliant career of Professor Arnaud Fontanet. And then instead of trying to complete this video, which I'm sure will be close to perfection, we will have a short exchange on issues of the time with regard to public health and epidemiology. So if we can start the video.
Pasteur Bicentenaire Award Selection Committee, dear colleagues, dear friends, I am deeply honored to be the recipient of the Pasteur Bicentenaire Senior Award. For me, Pasteur is a name that resonates with excellence in research, infectious disease, and global health. I am therefore incredibly proud to have contributed to the work of Institut Pasteur for the past 20 years. During this time, my team and I have studied transmission of infectious pathogens and clinical consequences of infection across the world, in Asia, in Africa, in the Middle East, in the Caribbean, and in the Pacific Islands, where the work on Zika, for which I have been awarded this prize, has been carried out. Together with our colleagues from Institut Louis Malardé in Papeete, we have linked Zika virus with severe forms of paralysis in adults and microcephaly in children born to mothers infected during pregnancy. While a lot of my work over the past 20 years has been done overseas, the last 3 years have been dedicated to the national response to COVID-19. It has been a striking reminder of how devastating infectious disease can be to our lives and our economies. With this prize, my team and I will continue to work on understanding emerging infectious disease. Specifically, we will seek to decipher the early public health response to the COVID-19 crisis, what went right, what went wrong, in an attempt to be better prepared for the next pandemic. We will also continue our work to study the transmission of pathogens at the animal-human interface, where it all begins. I would like to thank you for your recognition and generous support of this important area of work.
So, Arnaud, if you want to join us.
Arnaud, congratulations. I think Louis Pasteur and Charles Nicolle, if I may add this other name, which I consider the father of the concept of emerging infectious diseases, would be proud of you. I just would like to ask you a couple of questions which might help illuminate some aspects of epidemiology and its application to public health. And it's essentially to recall on your experience during the COVID pandemic. My first question is what do you think we have learned during this pandemic that will help improving the future of interventions against the next one?
Thank you very much, Philippe. Thank you, Christophe. Just to remind, I'm incredibly honored and to be here at this place. Difficult to summarize in a couple of minutes questions you were mentioning. What I think is, as an epidemiologist, have been real breakthrough during the COVID-19 pandemic. I would name two different areas. One of the mathematical modeling of infectious disease. For the first time, as far as I know, in a pandemic, mathematical modeling provided the tools for projections that have been incredibly helpful to governments when making painful decisions. And here I would like also to thank the work of Simon Cauchemez at Institut Pasteur, Vittoria Colizza at INSERM, and many others. I think it was really an incredible contribution to the way the pandemic has been handled. Another area as an epidemiologist that I think has progressed a lot during the past 3 years is the linking of data across different databases, the one on cases, the one on hospitalization, the one on vaccines, that have allowed real-time evaluation of the efficacy of vaccines. And that, again, has been, as far as I remember, the first time we were able to produce within a couple of weeks results that have been essential in evaluating how powerful these vaccines have been. So that would be, for me, the two great advances of the past 3 years, and I just hope we can continue in that direction.
Okay, thank you. My other question would be, at the beginning of the pandemic, I mean, in the first months, first wave, for instance, I mean, there were clear discrepancies between the impact of the pandemic on the population, I mean, both in terms of health and socioeconomic consequences. Can you, in retrospect, try to analyze the reasons for these discrepancies?
What I will say here is, if you look at Western Europe, where we basically have relatively similar population with terms of demographic structure, the health systems, governments, we still have, when we look back 3 years later, a ratio of 7:1 in the excess mortality when comparing countries like Italy and the UK and Denmark. And here, I think it's really important to understand why these differences exist. If I were to point at one aspect which I think has been crucial to understand these different responses is how fast governments have acted. And here, when it comes to Denmark, it is important to remember that the Prime Minister came on TV on the 11th of March. At the time, there were only 10 hospitalized cases in the country, to ask for the population to start not the lockdown, but really major restrictions in contact. And having acted so early has allowed Denmark not to fill the hospitals. Therefore, they were able to move out of lockdown very early. They repeated that for the second wave in the fall 2020. And I think these very precocious measures were the way they preserved not only human lives, but also economies. And for that to happen, you need an incredible level of trust between scientists who advise the Danish Prime Minister and the population. When the Prime Minister came and said, we will ask you to start to restrict your contacts, even though nothing was visible in hospitals. What I'm a bit afraid at the end of this pandemic is that this trust between population, scientists, and government has been very much challenged with all the disinformation we have seen, and that we have to rebuild it. And I hope with time we will be able to recreate this link that will be essential if we have to face future crises of this kind.
Okay, so in no way the good situation in Denmark was related to beer consumption.
That was not planned in advance, I have to say.
Okay, so I think for the sake of time, we will stop here, and I think we can go for a big round of applause for Arnaud Fontanet.
So thank you, Philippe and Arnaud, and congratulations, Arnaud. So we'll now move to the Junior Scientist Pasteur Bicentenary Prize, and this prize is going to be awarded by Jeffrey Almond. So he was Vice President and Global Head of Research and External R&D at Sanofi Pasteur. And he's currently a professor at the University of Oxford, and he's going to award this second prize. Jeffrey.
Oui, merci. Apparemment, je dois parler anglais aussi. C'est difficile pour moi. Excuse my Yorkshire accent. It was an honor and a privilege to be a member of this jury. The level of the scientific contributions was amazingly high, particularly for the Junior Institute Prize. I would like to give all of the candidates a prize, but I can only give one person a prize. The rest of you get the beer, which is a really good second prize, and a very special beer, apparently. So the prize, in fact, is going to Melanie Hammond, and a moment. And she will present her video in just a moment, but I have to say it's a very nice area of work where she's been looking at the really early interactions between bacteria and the epithelial cells when they arrive in the body, and the signaling between the bacterium and the cell, and the implications of that signaling to the level of either commensalism or pathogenesis for the bacteria. And understanding that process, I think, is profoundly important for understanding our relationship with microbes. More of that perhaps in the question time. But I think first we should have the video. Thank you.
Good evening. My name is Mélanie Hamon, and I lead the lab Chromatin and Infection at the Institut Pasteur in Paris. In the laboratory, we study how bacteria modify host cells during infection, and specifically how they affect DNA and its organization through mechanisms that we call epigenetic. We focus on one bacterium in particular, Streptococcus pneumoniae, as we find it particularly interesting that it has the dual nature of being both a natural colonizer and a virulent pathogen, which allows us to study the balance that exists between the two. In one of our recent findings, we were able to show that a colonizing asymptomatic strain of Streptococcus pneumoniae was not just passively carried by host cells, but was actively modifying them to maintain homeostasis and avoid dissemination. And how very fitting is it that it is Louis Pasteur himself that discovered carriage by Streptococcus pneumoniae. This is a bacterium that he called the bactérie de la salive, or salivary bacteria. So it is a great honor for me to receive this prize tonight. And as a trained microbiologist, it is one of the greatest honors to be considered as carrying on the Louis Pasteur spirit. And I wouldn't have been able to do any of this without my fantastic team, Michael, Tiffaine, Matthew, Julia, Marianne, Justine, and Christine. I'd like to continue by thanking the selection committee for having chosen our work, and this prize will allow us to continue springboarding towards new research. I'd like to thank my friends and family and colleagues that are present here tonight, and all of you for your presence, and enjoy the ceremony. Good night.
So Melanie, if you can join us.
So congratulations, Melanie.
Thank you very much.
I liked your video very much.
Thank you. It was a difficult exercise.
I have several questions, but we have seen in recent years a reluctance, a difficulty in arriving at new antibiotics and dealing with the problem of antibiotic resistance. I wonder whether in your mind, understanding these early interactions offers new ways to intervene that might be more direct and more attractive to the industry in terms of developing new interventions to prevent pathogenesis. Would you like to just give your perspective on that?
Yes, absolutely. So this is a field of research that we actually are going into and we are exploring. In fact, if we believe that if we can understand the mechanisms by which bacteria are exploiting host cells, that we could then target those mechanisms as they are giving an advantage to the bacterium. And if we take that advantage away, then the immune system would then have a higher advantage and maybe help clear the bacterium. So I don't think it would replace antibiotics, but it would probably maybe could be used at the same time and help antibiotic treatments. Or offer new perspectives, yes.
And to what extent does this work help us to understand better commensalism? One of the things we've learned a lot in the last 10 or 20 years is the incredible complexity of the microbiota that live on us and in us. Yes. And understanding how those organisms arrive at homeostasis with us and with all of the animals and plants even around the world, it seems to me is a very important question. So this work takes us there?
So it's clear that that colonizing bacteria or microbiota, bacteria present in the microbiota are actually doing things to the host cells, and I think that that is definitely an area of research to go into to see what the cells are responding to, because this is how they are being carried. And I think it's not just a passive carriage, that there is something active there that we need to dig into and understand. Yes.
So the last time I was at the Institut Pasteur, we talked about how 200 years ago the bacteria were seen as the enemies that we needed to fight. And our perspective has now changed, I think. We now need to regard the microbes as sometimes our friends that are important in the balance of the ecosystems around us, and we have to protect them, not just fight them. And I was particularly interested in your work from that perspective. But thank you very much again. Congratulations again.
Thank you very much.
I think I have a gift
Thank
for
you to everyone. Thank you.
Thank you, Jeff, and congratulations again, Melanie. So the third prize has been awarded to a scientist in the Pasteur Network, and I'm pleased to invite Françoise Barré-Sinoussi. I know the Pasteur Network is dear to your heart. Obviously, you're known for your discovery of HIV and being awarded the Nobel Prize for this discovery, and I don't think I have to introduce you more than that.
Thank you. Thank you very much, Christophe. First, I would like to thank Pascale for asking me to be part of this jury. It has been really a pleasure. Secondly, I must say that honorary president of the Pasteur Network, of course, I'm very pleased to call the laureate of the Pasteur Network for this prize, Leo Poon. Leo Poon will come to the stage after the video, but I would like to say that it was very easy to select this prize because Leo is a virologist, so a second reason why I was very happy as well. But he's very well known for his work on respiratory infection, influenza, but also coronavirus. Leo has been associated to the discovery of the first coronavirus in 2003, but the reason why he is receiving this prize today is his work showing transmission of the SARS-CoV-2 from pet hamster to human. So he probably will say a few words about his work in the video. So I like to see the video now.
My name is Leo Poon. I'm a co-director of Hong Kong Louis Pasteur. I'm also a professor in School of Public Health, the University of Hong Kong. I'm a molecular biologist by training. I've been researching on emerging infectious diseases for over 20 years. Zoonotic viruses, such as coronaviruses and influenza viruses, are my major research topics. My team developed molecular diagnostic tests for these viruses. We also studied their ecology, transmissions, and pathogenesis. My ultimate goal is to use scientific findings to inform public health policy. In 2003, I had the privilege to work in our multidisciplinary team to study SARS, and I managed to discover the very first SARS-CoV-1 sequence. In the current pandemic, my lab is a WHO reference lab for COVID-19. We developed molecular diagnostic tests for SARS-CoV-2 in January 2020. We shared them with over 170 laboratories around the globe. We subsequently conducted many studies to understand the biology, immunology, and transmissions of these novel diseases in the last 3 years. Findings from these investigations, together with those by many others, assisted the development of evidence-based control measures against COVID-19. The Louis Pasteur Bicentenary Prize recognized achievement that captured the spirit of Pasteur. It is my great honor to receive such a heavyweight recognitions. Although COVID-19 is transitioning from a pandemic to an endemic disease, there are still many animal viruses which may have the potential to infect humans or even can cause the next pandemic. With this prize, I will continue to offer my best to contribute in scientific investigations under the One Health concept so that we have a better preparedness plan for the next.
So Leo, if you want to join us.
Congratulations, Leo, to you and your teams for this work. I have maybe 2 questions for you. The first one is regarding, the variability of the SARS-CoV-2. What is next? What is next? It's a very difficult question, I know, but what is next in human for SARS-CoV-2?
I wish I have an answer for that.
Right. I know you don't have the answer.
but I think the lesson that we learned from SARS, H5N1, and now SARS-CoV-2, bring in the issue that animal viruses can jump into human. We should able to try to work on that, to try to understand the ecology and try to identify the potential risks so that we can have evidence to try to reduce the risk further. So, we are human, so we cannot fight against the mother of nature, but we can able to modify it and reduce the risk. So I would urge for the One Health concept, try to understand those animals which have a close contact with us so that we can able to identify the potential risks and try to do mitigations.
My second question is related to your paper on transmission from hamster to human. What about the origin of the infection in hamster?
It is a good question. So I think we should not only look at the hamster. Now, the real problem right now is, although we found evidence to show that the hamster could transmit the virus to human, and then we can able to monitor this. These are pet animal. We can able to monitor it, but the danger will be something in the wild, right? I think the wild animal like the white-tailed deer, and they have been infected by SARS-CoV-2. So, and then also there are some mink which are able to be infected by SARS-CoV-2. And then, so to me, there are some animal species which may not be under our radar all the time. So I think we should urge for better surveillance on those unknown.
Okay, congratulations again. I think, Leo, that you are a real fantastic example of what can be done in the Pasteur Network. So congratulations to you and your team.
Thank you.
So the next prize is awarded to junior scientist who is an alumni of the Institut Pasteur. And this prize will be presented by Professor Staffan Normack. Staffan is a medical microbiologist. He was a professor in Sweden and the US. He's a member of the Royal Swedish Academy of Science and a member of the Nobel Foundation.
So I'm very proud to be here tonight and celebrate the Louis Pasteur Awards. So I'm honored to present to you that this year's Pasteur Alumni Junior Prize has been awarded to Professor Serge Mostovi, who is Professor of Cellular Microbiology and at the London School of Hygiene and Tropical Medicine. So before the video, I can just summarize a little bit of Serge. What he did was to discover something. He discovered that host cells can trap intracellular bacteria in cages, much the same as Frodo was in cages by that giant spider. It's a very similar story. But we will hear more from Serge about that story. So we take the video.
My name is Serge Mostovi. I'm a professor of cellular microbiology and Wellcome Trust Senior Research Fellow. My lab is based in central London at the London School of Hygiene and Tropical Medicine. The school is a research institute focused on infectious disease and global health impact, and in this way captures l'esprit Pasteur. It is an exciting time. I encourage you to come visit us. In my lab, we are focused on the cell biology of Shigella infection. Septins are a poorly understood component of the cytoskeleton, best known for their role in cell division. What we discovered is that septins can entrap Shigella in cage-like structures and target them to destruction by autophagy. It is now of major interest to understand the underlying molecular mechanisms and to validate these events in vivo using relevant animal models. For this, we developed the zebrafish as a new animal model to study Shigella infection, and in this way discovered new roles for septins in host defense. Collectively, the results generated from this research program can provide fundamental insights into cellular immunity. In my lab, we are primarily focused on septin interactions with gram-negative bacteria. I will use this prize to understand how septins can interact with gram-positive bacteria. This research avenue has the great potential to illuminate the breadth of membrane proteins that septins can interact with, and may even suggest the evolutionary origins of septin bacteria interactions. It is hoped that this information can provide important clues towards understanding bacterial disease and may even suggest novel therapeutic strategies.
Okay. So we ask Serge Mostovi to come to the stage. So it's great to have you here and really honored to read your work and to understand what you have discovered, because we have never met before. I have a few questions to you, because you have really made a discovery. You have shown a new system, how the host can engage themselves with intracellular bacteria. And you say that you have studied this mainly now in gram-negative bacteria, and that the shape and the form of the bacteria can play a role. It seems to be that these septins interact with the pole of the bacteria. And we know that gram-negatives have an outer membrane, so that is lacking in gram-positive bacteria. And as you know, gram-positives are quite often cocci, so they are only a little pole or two poles. So what are your thoughts about gram-positives and how they can the host recognize them to form cages or not?
Fantastic question. Thank you very much. I first want to say it's very special for me to receive this award. I spent transformative years here as a postdoc in the lab of Pascale Cossart, and it really changed, the way I tackled infection biology. We made some special discoveries that led me to the reason that we've obtained this prize here today. And what we're here to discuss is, we discovered a new way in which host cells can recognize intracellular infection. It is really primarily based on the way bacteria are shaped. And so septins are a cytoskeleton component that like things on the order of 1 micron in diameter. So to come back to your question, if your gram-positive bacteria is on the order of 1 micron in size, septins will recognize it. Now, the cell surface plays a major role, and that's what this money will go towards, the understanding the bacterial factors that can either invite or evade septin recognition. But we know already that shape plays a major role, and not all bacteria want to be recognized by septins, so it's of great interest to us to catalog these effectors.
What is your thought? I've always thought that when things are forming, let's say, filament around the bacterium, that the first initial contact is very central. Because I'm thinking about this as a kind of that the host that the bacteria has some nucleating function or changing the shape of that septin so it can change shape and then form a filament similar to what we see in amyloid type of conditions. Is that something you're thinking, or is it something totally different?
No, it is not totally different. I think what septins may be able to do is to recognize that initial event of polymerization and to prevent, perhaps, the filamentation process of which you discuss. One thing that septins do very well is they recognize actively dividing bacteria, and they are very efficient at stopping bacteria from dividing. So perhaps in different changes when bacteria do try to change shape to avoid septin recognition, I think if I do my job well, perhaps we can tweak the septin cytoskeleton to recognize bacteria of different shapes of which you speak.
Okay, I think you have a great future. I think it's a great discovery, and I'm really congratulating you to this prize.
It's a great pleasure. Thank you.
Congratulations.
Thank you, Stefan, and congratulations again, Serge. So we are reaching the award of the last prize, which is going to a senior scientist, an alumni of Institut Pasteur, and the jury member who is going to award this prize is Professor Alain Fischer. He's a pediatrician, he's a founding member of the Imagine Institute, professor at the Collège de France, and is the new president of the Académie des Sciences in France. Alain.
Thank you. Good evening to everyone. I have to say I'm also very pleased to have been part of this jury and enjoyed the discussion, and we had a good one about this fifth prize. Just a few words about Bruno Canard, who is the laureate. Bruno Canard is a directeur de recherche CNRS on the campus of Marseille Lumini, a very nice place. I'm sure many of you know the place. In an institute named Architecture et Fonction des Macromolécules Biologiques. He spent some time at the Pasteur Institute a long time ago when he did his PhD with the present director, Stewart Cole. So I think to know better about him, we should listen to what he wants to say to us.
Hi, my name is Bruno Canard. I am a director of research at the French CNRS in Marseille. And first of all, I'd like to thank my mentors, my teachers, all my colleagues in the team, and postdocs and students who have all contributed to my research over these past 25 years. I've been working on RNA virus enzymes, enzymes from emerging RNA viruses mostly, and my kind of obsession has been to understand how this enzyme work. To do this, we have to understand their structure, we have to understand their mechanism of action, and how they are coordinated one with each other. This prize will help me to explore and address questions that are not easily supported because they are sometimes high risk and out of the beaten path. And I'd like to address in particular how the viruses and the replication enzyme get their building blocks, their nucleotide, how they are fed with it actually, and how they are using them to synthesize genomes, RNA genomes of long RNA viruses, such as that of the coronaviruses, for example, and how in the end this yields an infectious viral particle. Thank you.
Bruno, congratulations. This is really nice. I'm not a virologist, so pardon me for the questions I may ask. But I would say, among the many things you have done in structural biology of viral proteins involving enzymatic activity, I'm pretty impressed by the work you have done with the evidence that there is an RNA repair pathway that is used by viruses like coronaviruses. So I'd like to have your thoughts about what does it mean for a virus to have such a system that if it works too much, obviously they will create a problem, but if it doesn't work at all, there won't be any selection. So could you elaborate a little bit more on this?
So it was a surprise to see at first, then when I came into the coronavirus field, that there was a sequence signature of exonuclease and endonuclease, an enzyme that actually cut RNA. So the problem was to understand why, how enzyme could actually be not cutting their own RNA. And it became clear that this was related to the size of the virus, and the coronaviruses have a very long genome. So it became very interesting to see what was the load of the enzyme. Was the enzyme working a lot, correcting a lot of mistakes, or in fact very lazy? And we could actually see that the enzyme has evolved a manner to go very, very fast and synthesize the genome and be very promiscuous. And in that, it does make a lot of mistakes, and then the exonuclease is there to correct. And this is actually linked to the evolution of these long RNA genomes. And it's a fascinating field, I think, that will yield a lot of other surprises.
Thank you. Another obvious prolongation of your work is to foresee new treatments, new antiviral drugs that we need for coronaviruses and potentially for others. So you have been involved in testing and developing such drugs for coronavirus, SARS-CoV-2. So do you think today the methodology and the way to go based on the basic knowledge of how the virus cycle goes is making progresses that we should be optimistic that for the next pandemics we'll have soon, very rapidly, the good antiviral drugs?
This is a message that I've been trying to, with others of course, trying to implement and introduce, that we need to be prepared in advance. And I will give only one example. When we have this variants coming, it was a surprise to see that Omicron, for example, had at first like 60 mutations. And we know how to interpret 30 of them in the spike. And I say bravo to Olivier Schwartz in Pasteur, for example, who has been very active in the field. But we don't know very much about 30 other mutations. And they are doing many things that we don't understand. And why we don't understand that? It's because we lack basic science on that. We lack structures, we lack mechanism, we lack traditional biochemistry, enzymology. And I really hope that studying this type of academic science and fundamental science in advance will really help to prevent, or at least to be much more prepared for the next pandemic.
For sure, we'll need you for the next one. Thank you very much, and congratulations again.
So thank you very much. So before we close this ceremony, I'd like to invite Professor Emmanuelle Charpentier to give her concluding remarks. So you know Emmanuelle for work on genome editing and having been awarded the Nobel Prize in Chemistry together with Jennifer Doudna in 2020. She's the head of Max Planck Institute for Infection Biology in Berlin. And interestingly, she's been working at Institut Pasteur, where she graduated as a PhD student. So, Emmanuelle.
Thank you very much. So, dear Stewart, Christophe, dear Pascale, dear laureates, dear members of the jury, dear distinguished guests, dear Pasteuriens et Pasteuriennes, dear colleagues and friends. Actually, I was told before the ceremony that today was the last event of the series of events that were organized to celebrate Monsieur Louis Pasteur. So I believe it's a good time to thank the Pasteur Institute, the Pasteuriens and the Pasteuriennes, all of you and all of those really contributed to further highlight macrobial research. I guess this is what Louis Pasteur was doing, and to celebrate, more Louis Pasteur and his great achievements. I would like to thank the 5 laureates. So Arnaud, Serge, Mélanie, Bruno, and Léo, for presenting us very briefly the focus of their research. And congratulations for your great achievements. For sure, we should never forget the researchers who competed for the prize, are not laureates, but are still nevertheless doing great research. And we are always, pleased to see the commitment and hard work of scientists. As it was said, I had the privilege to receive my pre-doctoral and doctoral education at the Pasteur Institute. This was about 30 years ago. This was another time. The Pasteur family was different. It was, I think, smaller, maybe less complex with regard to administration and bureaucracy, like everywhere in the world. Actually, I was studying antibiotic resistance and mechanisms explaining antibiotic resistance in the bacterial pathogen Listeria monocytogenes, not in the lab of Pascale Cossart, but in the lab of Patrice Courvalin. And I would like to mention here, this is the purpose of me saying this, is just that as an ex-Pasteurian, I belong to the large number of ex-Pasteuriens and ex-Pasteuriens who have, worked in a way as ambassadors, all over the world. I think it's about trying to transmit the Pasteur spirit, the esprit Pasteur, to the members of your team, whether you succeed or not, and also to your colleagues, So at least my experience as a microbiologist abroad is that it's maybe easier to defend microbial research in an environment at the Pasteur Institute. It may not be always easy to defend microbial research with regard to funding agencies, even for the Pasteurian and Pasteurian. But it's not always easy to defend microbial research abroad in institutions that are not really focused on microbiology. And independent of this, there are also, a research that is not microbial research done at the Pasteur Institute. And I think it's very important to highlight that a number of scientists carry on with them, whether it's for the purpose of microbial research or other type of research, carry on with them the Pasteur spirit. I'm not sure that people appreciated always when I was always coming with when I was at the Pasteur Institute or Pasteur, etc. But at some point they understand that they have to accept. We are facing challenging times, as you know. I think at least the COVID-19 pandemic allowed us to highlight how important fundamental research is. How important it is to carry on with the research on microbes, including pathogens, that, as it was said, we feel quite humble and, when we face Mother Nature and the power of microbes that have largely evolved to have fantastic strategy to trick us and change the world. It also reminded us, nevertheless, at least for me, how international, global the research has become over the past 15, 20 years. Very competitive, great developments of wonderful technologies. We feel, I guess, lucky to have the chance that vaccines were developed, so fast. And this is always with the contributions of large number of scientists, even those who may not be directly behind, the development of a vaccine. You have always, when you have a discovery, you always have the involvement of a large number of scientists who have contributed to the puzzle. And the Pasteur Institute is highly recognized for the excellence and the research performed in the microbiology field. What COVID-19 as well, the pandemic has shown us is to which extent also the private sector has changed and has evolved also with regard to research and development. You have a large number of biotech companies now doing also very interesting work. Which leads me to also the challenges of the science in today's world. That is that we certainly, at least in France, it's maybe not the case in Germany, but in France, there is really a need to improve the funding of public research. I think it's critical and it's really extremely needed in emergency, I would say. With the Pasteur Institute being not only a public institution, but also a mixed public-private institution, there is also the great importance of the funds from donors and philanthropists. So it may not be so much, let's say, a culture in Europe in contrast to the US. that benefit a lot from funds from philanthropists, but I wish that philanthropy would play a higher role in supporting the research that cannot be obviously fully supported by public funds. The scientists really rely on more funds. I mean, science became more and more expensive. It's great developments of technologies that are extremely extremely expensive, but also that evolve extremely fast. So everything relies on really more and more important funds. The challenges also are associated to the complexity. I mean, not only in science, as you know, but specifically also in research and in specifically in research environments in the public sector. I think the past years have witnessed, let's say, a development of more complex systems with too much bureaucracy, maybe too much importance on an administrative aspect that may not be always efficient. At least this is my experience, obviously not in France. And I think it's really critical to really think of the systems, try to streamline them, bring more simplicity. Because we have an issue in our days is that the young generation tend to be more attracted to the private sector. That is quite strong in certain countries. And we see our young scientists, which we train, leaving the public sector for maybe good reasons, actually, I don't know. Not always good reasons, but also good reasons. And it's not easy to carry on with competitive research, at least in Europe and also where I am. It has accelerated the past 2-3 years. And so I think it's a challenge, and which tells us that we need to also think of systems that have to evolve to be able to be attractive for the generation of young scientists, which we need tremendously. As you know, the future of our world and society relies mainly on science and technologies, and specifically in what is, I think, mostly the goal of the Pasteur Institute, carrying on research in the medical sector, at least as an orientation. And I would like to finish with a quote from Louis Pasteur. There are lots of quotes, actually, from Louis Pasteur, but there is one that is, "A chance favors only the prepared mind." And I like this quote because this is always a challenge as a scientist, whether you can carry on with your research. Obviously, the ideas are not lacking, but you have to find nevertheless something interesting, and there are so many aspects you cannot control. But I think there is an aspect that you can control, and this is also what I learned actually at the Pasteur Institute is you have human beings behind it. Research is where it is now because we are scientists who are, giving, I think, a lot of ourselves. Drive is important, commitment is important, the sense of empathy for what we are doing. Actually, I believe at least we are working hard. And I think sometimes it's all about, simple things as, l'amour du travail bien fait. And I think when we have this in mind, we can actually do great science. Science is also, this is what I like to be, at least in the environment of scientists. It's one meets so many people. It's, one can have a lot of fun in addition to a lot of frustrations. And I think this is, I'm sure, also what you find at the Pasteur institute, which also evolved to be more international, to have a large number of exciting young scientists and group leaders. And personally, I wish a great success to the Pasteur Institute, and I remind all of you, all of those who wish to support the Pasteur Institute financially or by other means to do so, and to also support research in general, because we desperately need support. Thank you very much.
Okay, so we've reached the end of the ceremony, and I'd like once again to thank the jury for their hard work. I'd also like to congratulate the alumni once more. I'm going to invite them onto the stage so we can have a group photo, and while we're doing that, I would like to invite the audience to go and celebrate Pasteur's work on fermentation at the reception, and we will join you shortly. So, thank you very much.